Ellen Shrock from the Elledge Lab at Harvard. As I understand, Ellen just recently successfully defended her thesis and has a incredible number of important publications already. And so we can say Dr. Shrock, and we'll let you get into it.
Okay, thank you so much. Yeah, so I actually first heard about the AIR from Ramit Mehr at a Keystone conference back in 2018. So I was very honored to be invited to speak to this community today. So the title of my talk is Germline-encoded Amino Acid Specific Binding Motifs Drive Immunodominant Public Antibody Responses. So what I'm going to be telling you today is published, so it's available online. So just to give a brief vignette of what I've been studying, we noticed previously that if you look at all of our antibody responses to SARS-CoV-2, as we've all been exposed one way or another, there are regions of the spike protein where over 70% of us make antibodies to those regions. And so this is very striking that so many different people in a population are making these recurrent responses to the same region of a protein. And this was kind of what initiated my project. So previously, the Elledge Lab developed a method called AIRR-C to identify targets of human antiviral antibodies. And so the way it works is that there's a phage display library, and each phage encodes, each phage displays one viral peptide on its surface, and that viral peptide is encoded within the genome of the phage. And collectively, this whole phage display library spans all the proteins from all of the viruses known to infect humans. So it's meant to be a comprehensive synthetic representation of our human virome. And then if you mix this library with a drop of blood, which contains circulating antibodies from all of a person's past viral exposures, and then identify which phage are recognized by the antibodies in the blood, then you can identify the targets of human antiviral antibodies. So this is done with a next generation sequencing readout. And since one of the inventors of phage display is here, I have to shout out that this method is of course indebted to the work of George Smith, Gregory Winter, and Jamie Scott, who's here, which is very cool. So after running these AIRR-C experiments and profiling the targets of human antiviral antibodies for hundreds of people, one thing that people previously observed was that the immune response is remarkably reproducible across individuals. So here, each column is a different person, and this is a diverse cohort from 4 different continents. And each row is a different peptide from, in this case, respiratory syncytial virus glycoprotein G. So when these people are exposed to this virus, they all, or many of them, make responses to one particular peptide. So their immune response tends to focus on one particular peptide within this viral protein. And it's recognized by all these people. And here's another example of one of these public immunodominant epitopes. In this case, it's from adenovirus-C pentin protein. In some cases, there are public epitopes that are less frequently recognized, but still recurrently recognized. So there is a range in the frequency with which a public epitope is recognized. But in total, we found 400 examples of these immunodominant public epitopes from 62 different viral species. So this seemed to be a general phenomenon, not specific to any virus, but just a general phenomenon. And I don't have time to show it today, but we actually mapped which exact amino acids are recognized within these peptides, and very frequently different people are recognizing the exact same residues within these public epitopes. So the question was, we all know that the naive antibody repertoire is extremely diverse. So why are there shared antibody responses across individuals? And this is really a question of immunodominance. So why does the antibody response prefer to recognize some targets over others? I'll give a bit of background first. So yeah, one of the first questions that I asked was whether antibodies that recognize public epitopes are conserved across people. So I'll just describe the structure of an antibody. Of course, this community does not need the background, but for any viewers on YouTube, this may be useful. So antibody diversity is generated through VDJ recombination, which is a rearrangement process that occurs in each B cell during its maturation. So in the germline locus of the antibody genes, there is an array of many different V segments, followed by many different D segments and many different J segments. And this is an array of gene segments. So the array itself is pretty similar across people. There are some allele polymorphisms for a single V gene segment, but overall, the sequence of the array is pretty similar across people. And each V gene segment has a certain name. So for instance, one might be called immunoglobulin heavy V12 or immunoglobulin kappa V15 or immunoglobulin lambda V140. So that refers to the heavy chain V or the light chain. And there are two light chain types, kappa and lambda, and each antibody either has a kappa or a lambda light chain. So during BDG recombination, 1V, 1D, 1J are rearranged and recombined together to make the functional antibody. And this is the part of the antibody that makes contact with the antigen. And a very similar process happens in the light chain, although it's not shown. And again, there are two different light chain types, lambda and kappa, and this will come up later in the talk. So within the recombined functional antibody sequence, there are some particular regions called the CDR1, CDR2, and CDR3. And these are loops that stick out of the antibody in the sequence and are available for making contact with the epitope. So here's the light chain CDR1, CDR2, and CDR3, and the heavy chain CDR1, CDR2, and CDR3 loops. And so the CDR3 is by far the most diverse part of the antibody because it contains, it spans the end of the V through the D through the J. And so it contains both combinatorial diversity as well as junctional diversity because mutations are generated at the junctions. So it's by far the most diverse part of the antibody. And in contrast, the CDR1 and CDR2 are encoded within the V gene. So they're encoded by the germline V gene. And so their sequence in the final antibody remains pretty close to the germline V gene sequence. And in total, there's around 10 to the 13th different possible antibodies. So the CDR3 has typically been thought to contribute the most to specificity because, in particular, the heavy chain CDR3 is thought to contribute the most to specificity because it is the most diverse region of the antibody. So this will come up again later in the talk. So back to the first question, are antibodies that recognize public epitopes conserved across people? So to answer this question, I took a few example public epitopes and I used them as baits to stain memory B cells from many different people. And then I sequenced the memory B cells to get their heavy and light chain sequences. So this was the public epitope that I used. It was from influenza A hemagglutinin. And I found 9 different antibodies from 7 different individuals. And what was interesting and unexpected was that they had very different heavy chain CDR3 sequences. So this region that's thought to contribute the most to specificity actually showed no conservation in sequence, suggesting that it might not be so important for recognizing this public epitope. But in contrast, the V genes in both the heavy and in the light chain were perfectly conserved. So this one heavy V551, which is one choice out of approximately 40 different possibilities, and kappa V41, which is one choice out of around 70 different possibilities, were perfectly conserved throughout all of these antibodies from different people. And it was a similar story for another example public epitope, in this case from Epstein-Barr virus. So there was a strongly enriched V gene usage, both in heavy and light chain, but no observable conservation of heavy chain CDR3 sequence. So this suggested that recognition of the public epitope might be mediated through the V gene, perhaps through the CDR1 and CDR2, rather than the CDR3, since the CDR1 and CDR2 within the V genes were conserved, but the CDR3 was not. So the second question I asked was whether public epitopes have skewed amino acid composition. So I looked at the amino acid composition of public epitopes recognized by kappa antibodies with kappa light chains, public epitopes recognized by antibodies with lambda light chains, and as a baseline, the amino acid composition of the entire human virome library. So that's what I'm showing here. And in red is the composition of the kappa public epitopes, or public epitopes recognized by antibodies with kappa light chains. In blue is the composition of public epitopes recognized by antibodies with lambda light chains. And in gray is the composition of the entire human virome library. And the thing that struck us the most was that lysines are extremely enriched in lambda public epitopes, but not kappa public epitopes. So there were other things that were significant as well. For instance, prolines were enriched in all public epitopes. Serines, threonines, and valines were depleted from public epitopes, but the lysine enrichment in the lambda public epitopes was by far the most significant finding. So then we asked whether lambda V genes might have some germline-encoded affinity for lysines. So kind of reminiscent of an anchor residue for MHC and T cell receptors. So, or actually peptides and MHC, sorry. But the way I answered this question was that we analyzed all the structures of human antibody-antigen complexes from the protein data bank. And we subsetted the structures by which V gene they contained. And then for each residue of the epitope, we annotated every antibody residue that it contacted. And then we searched for recurrent interaction motifs. And then from this analysis, when we were looking at the lambda V genes, so looking at all the structures that contained lambda V genes, we got results like this. So for one lambda V gene, this is lambda V657, we saw that there were 6 different unique PDB structures recognizing very different antigens. So flu, some autoantigens, spike, HIV, so very distinct antigens. And in each of these cases, there was a recurrent motif in the antibody that was used to bind one specific residue in the antigen. So in this case, the antigen residue that was contacted was a lysine. So this is the antigen residue, and then here are the antibody residues that contact that lysine. And so we saw that in every case, there was a tyrosine at position 32 and aspartic acid at position 51 that were used to contact this lysine. And in this column, we checked to see how many of these residues were germline encoded. So germline encoded within the V gene. And we saw that indeed the tyrosine and the aspartic acid were both germline encoded. So essentially, this V gene seems to have a motif that is primed to recognize lysines. This is showing the structures of all of those PDB identifiers that I showed on the last slide. It's showing the same information. So there's recognition of an antigen lysine shown in magenta, and then the antibody residues are in cyan. So there's this tyrosine at position 32 that is making non-polar interactions with the lysine. It may be aligning the carbon chain of the lysine, and it's germline encoded within the CDR1 of this V gene. And then there's an aspartic acid at position 51, germline encoded within the CDR2 of this V gene, and it makes salt bridge interactions with this lysine. And this motif was recurrently used in all these different structures, all these different antibody-antigen structures. So we named this a germline-encoded amino acid specific binding motif, or a GRAB motif. It's germline encoded because it's encoded within the germline residues of the V gene, and it's amino acid specific because we only observe this motif interacting with a lysine in these structures and no other amino acid. And it's a binding motif because it is involved in the recognition of the antigen. And as we were expanding this analysis to every V gene, we noticed that there was a family of 6 different lambda V genes that share very similar GRAB motifs that all seem to be specific for lysine. So these are the V genes here, and they're 6 different lambda V genes, and their binding motifs have very similar properties. So the aspartic acid at position 51 is present in almost all of them, here and here and here and here. And this one has a bit of a different interaction where the aspartic acid is at position 52C, and then there's a pi cation interaction at position 51. And then the tyrosine at position 32, making the nonpolar interactions, was also very conserved throughout these different V genes. And in total, 75% of all the antibody-antigen structures on the PDB that have these lambda V genes exhibit this GRAB motif, using these residues to contact a lysine in the antigen. So if an antibody has one of these lambda V genes, it seems to very frequently use this motif to target a lysine in the antigen. And we think that these GRAB motifs likely underlie the enrichment that we observed in lysines in lambda public epitopes, but not kappa public epitopes. And I just want to mention that there were, I believe, 4 other lambda V genes for which there were no examples of structures in the PDB, but they shared conserved germline encoded residues that suggested that they would form the same GRAB motif. So that's as many as 10 out of 31 of our total lambda V genes seem to have germline encoded specificity for lysine. So then we asked whether other V genes, apart from this lambda preference for lysines, whether other V genes had GRAB motifs specific for other amino acids. And we ended up finding 18 V genes with GRAB motifs that seem to be specific for certain amino acids. This is an example of another one. So this is in heavy V321. This GRAB motif seems to be specific for aspartic acid and glutamic acid. So in this interaction, it's completely CDR2 encoded, and there are a series of germline encoded serines, some of which make hydrogen bonds with the aspartic acid, and then there's a tyrosine that may make nonpolar interactions. And So 4 out of the 8 unique PDB antibody-antigen structures that have this V gene exhibit this grab motif interaction to recognize a aspartic acid or glutamic acid in the antigen. So then we asked whether grab motifs are important for recognition of public epitopes. And I'm running out of time, so I'll go through this a little bit quickly. But we had a good example because our antibodies that we found to recognize the flu public epitope had heavy V551, which was one of the V genes for which we had identified a grab motif specific for a lysine. And lysine was also one of the critical residues of this epitope that we observed. So I took 2 of those antibodies and I mutated the residues of the grab motif. So each of these 4 residues of the grab motif in the antibodies to see what effect that would have. So I'll only show the data for one, but it was very consistent for both antibodies. So the wild type antibody binds to the peptide. This is a dot blot where you spot the peptide on and then add your primary antibody and secondary antibody. But in contrast, if I mutate any of those, any individual residue of the grab motif, then recognition is severely weakened to this influenza A public epitope. And in contrast, if I mutate some residues of the CDR3 or a residue nearby to the grab motif, frequently I don't see an effect. Then I also tested binding of the antibody to comprehensive mutants of the public epitope. So this is the epitope sequence, and then these are all of the possible amino acid substitutions. So it's a comprehensive mutagenesis, and I'm looking at the footprint that the antibody makes. So the antibody has some critical residues which are no longer recognized. When you mutate those residues, then the mutants are no longer recognized by the antibody, and that this creates an antibody footprint. So the footprints for these mutants, the non-grab motif mutants, were identical to the wild type pretty much. But for the grab motif mutants, the footprint changed. So recognition of the lysine was specifically abolished, and then there was increased dependence on the other residues. So this was evidence that the grab motif that we had identified from the protein data bank in structures that were not related to our public epitopes, this grab motif was indeed working in the antibodies that we found to recognize the public epitope that we had identified. And it was mediating recognition of a critical lysine residue. So the model of immunodominance that we are proposing is that if there is if what is needed to recognize an epitope is a very specific CDR3 sequence, then that's going to be a rare number of B cells that satisfy that requirement. And so that may lead to a private antibody response. But if one or more grab motifs can contribute enough binding energy and be sufficient to initiate an antibody response to an antigen, then there's going to be a much larger precursor population, and that can lead to a public antibody response. So we think that grab motifs may have evolved in order to recognize pathogens quickly and efficiently before you evolve a very specific rare CDR3 sequence. But they also play a role in viral immune evasion. And this has been demonstrated very clearly for SARS-CoV-2, where there are some public antibody responses that are neutralizing, and these are systematically evaded by SARS-CoV-2 in the viral variants. So it's possible that private neutralizers may be superior therapies because the virus encounters them less frequently in the population and has less of a selective pressure to evade them. And maybe vaccine strategies could be developed that already mutate those public neutralizing epitopes so that people start off making more private neutralizing responses. We think that there may be some decoy epitopes. So we did test whether any of those flu-specific antibodies were neutralizing, and none of them were. So it's possible that over time, a virus may conserve some public epitopes that are recognized very well by the human immune system, but are not but those antibodies are not neutralizing. So those could be decoy epitopes that kind of delay the evolution of more neutralizing antibodies. Yeah, and there could be some coevolution to recognize lysines, kind of this expansion of genes that are that have germline-encoded preference for recognizing lysines suggests that maybe our pathogens have an advantage to be enriched in lysines. And so yeah, maybe there is a benefit for viruses to be enriched in lysines. And I'll skip this for now. And we think there are many more grab motifs out there that are to be recognized. It's just a matter of finding the right ways to identify them. And I want to thank especially my mentor, Steve Elledge, who's been incredibly supportive of this work. And Richard Timms was helpful in coding part of the PDB analysis. And Tomasz Kula was my rotation mentor. And I'll take any questions.
Great. Well, thank you for a great talk. Before we have a few minutes for questions, I wanted to mention the next seminar will be September 28th with Peter Meisman. So we'll be taking a break for the summer. I really appreciate all these speculations that your work brings up and all these possibilities. Do we have the questions from the audience here? I've got a million, but so trying to remember him.
I think David. Oh yeah.
Yeah. Who do we have? Oh, David's got one. Yeah.
Yes, very nice talk. I'm very impressed by this
Here we go.
conserved V segments and totally random CDR3 sequences, because that's something we don't match in TCR often. If the V alpha or the V beta are specific, then we also have kind of a conserved CDR3 following. But I just want to follow up on your idea of benefits of having lysines in virus genomes or proteomes, which kind of fit with my story, because what we see is that lysine is not good for TCR recognition in the middle of epitopes. So who knows, maybe there's some aspects that also make them evading T cell recognition.
That's a good
Do you
question.
think it's something realistic, or does it fit your model?
Are you referring to the benefit for viruses, or are you referring to this being consistent with
the benefit for viruses to be enriched in lysines?
Yeah. So
could be also a way to escape T cell recognition.
I see. So we have been thinking about this a little bit differently. So for instance, in Omicron, what was kind of striking was that 8 of the 30 amino acid substitutions spike are mutations to lysine. And so my colleague has shown this in blue. So they do seem to be, many of them are solvent exposed and could be recognized by antibodies. And there was a modeling paper that I think was on bioRxiv and discussed in the New York Times, where there was a suggestion that maybe this enrichment of lysines in Omicron was a benefit because it stabilized the virus in aerosols, because the aerosols have mucins, which are highly sialylated, and those are negatively charged. So if the virus has more lysines, it could stabilize them in these aerosols and then promote transmissibility, for instance. So that was the hypothesis that we were thinking of, but it's interesting to hear that you also have this other hypothesis about evading the TCR recognition.
So
okay,
maybe go to Marcos.
Thank you, Ellen. Great talk. So I was wondering, so you're defining these public epitopes as immunodominant based on the fact that multiple people, or in some cases, like almost everyone, binds them, right? I was wondering about, like, do you have a sense within each individual how important they are, right? Because it could be that an epitope is recognized by everyone, but within each individual, like only a small fraction of antibodies recognize that. And I was wondering if you've looked at, whether those public antibodies are also important within people.
So do you by important, do you mean how the magnitude of the response?
some sense, yeah, I know it's hard to disentangle, right? The abundance of antibodies that bind each epitope times their affinity, right? Like the total contribution will be, I think, a product of those two things.
Exactly.
Right.
Yeah. So I don't think we have data on that. I think that we think that the same mechanisms are driving immunodominance and publicness, because precursor affinity and frequency is probably an important driver for both of those things. And these antibodies are present at a titer high enough that we can detect them in a very large fraction of the population. So that's also suggesting that they are immunodominant to some extent. There are very few cases where this has been studied actually in depth, but one of them was a paper maybe by Laura Walker, but it was a paper where they were describing babies' responses to RSV, and they found that there were two immunodominant responses, and actually one of them is to a public epitope and uses a grab motif in that interaction. So in that case study, it is certainly immunodominant, and it is also a public epitope.
Got it. Thank you.
Go to Jamie, please.
Yeah, thank you. This was a wonderful talk. It was really interesting. I wanted to ask you if you have any idea with the virus scan, this approach, what fraction of the total antibody response against a virus is really being picked up by these sites. In other words, you know, for the most part, you have discontinuous epitopes that don't have necessarily cross-reactivity with a linear peptide sequence on a given protein. And as a result, you know, it's really a tiny subset, what our lab had showed, that cross-react with peptides. So I was wondering what you guys are seeing there, and could that be one reason why you know, a lot of times these epitopes that are linear might not necessarily have anything to do with neutralization or clearance of the virus?
Okay, so is your question whether the conclusions are applicable to conformational epitopes as well, or whether
no,
grab motifs?
That when you're doing a virus scan, what percentage of the antibody response, or what fraction in general, do you think is actually cross-reacting with those, with your linear peptide?
Yeah, so I think, as you said, it's probably a tiny fraction. We had a collaborator who was finding monoclonal antibodies against spike, and he gave us, I think, maybe 50 of them to test in VRScan. We didn't find targets for any of them. So it's definitely a small fraction.
Okay.
Yeah, I mean, in that case of the RSV public epitope that I described, those are neutralizing, and they are also public epitopes. And so that is a linear epitope, and it's a neutralizing epitope as well. We also did, so previously we did, tried to map the epitope landscape of SARS-CoV-2 from COVID-19 patients, and we did find several epitopes within the RBD. So those are potentially neutralizing epitopes. Actually, that overlapped with the ACE2 binding contact sites with the RBD. So those were likely neutralizing epitopes that we were able to pick up with VRScan. So definitely we're picking up a small subset, but it may include neutralizing, probably does include some neutralizing epitopes.
Oh, for sure. Great, thank you.
Okay, so maybe one last question from Anastas, and then we'll wrap up.
Thank you, great talk. I'm really excited about your work. And I was just thinking, have you guys thought, are there any implications of your conclusions about these public epitopes with regard to our model for repertoire selection? So would it pose a problem for autoreactivity, or Are they positively selected actually? Have you thought about that?
Yeah. So, so with regards to autoreactivity, so we had a separate study where we were looking at, we were using the AIRR-C platform to look at antibody responses to peanut allergens in allergic people. So it was profiling the specificity of IgE in allergic patients to peanut. And we also observed public epitopes. So we think that this, and in the protein data bank, there are also examples of grab motifs recognizing autoantigens. So I would say that the ability to recognize public epitopes is just part of our antibody response, and it doesn't care about whether the protein is from a virus or a human. I think maybe because autoantigens should not be presented on MHC2, this could be a way to not provide T cell help to autoantibodies, and so they just don't proliferate. But in terms of the phylogeny and selection of grab motifs, so we did, I have looked into this a little bit. Some of these lysine specific lambda grab motifs do seem to be conserved. They're in cats and dogs and bats and cows, and they're not in mice, but that's probably because mice lost them along with most of their lambda V genes, but they are conserved in many other species, so they're very ancient, and I would think that they are conserved, yeah, selected for. I haven't done that full analysis for all the grab motifs, but I'm intending to.
Do you think that there will be the same frequency in the IgM repertoire and the IgG repertoire, or there may be differences?
You mean antigen selected versus naive?
Yes.
So I have done this analysis with data from the observed antibody space database, looking at the V gene composition of the antigen selected repertoire versus the naive selected repertoire. It does seem to be quite different. There are many, many things that are significantly enriched in one versus the other. Yeah, I didn't see specific enrichment in the lambda V gene, the lambda V genes that have grab motifs, but maybe that's because they're slightly redundant. I'm not sure, but yeah, there are many differences.
Thank you.
Went a little over with the questions, but I'd like to thank both speakers for great talks. And as usual, Pam Borgart for organizing everything and keeping us in line. And again, the next talk is going to be in September. We'll take a summer break from the webinars. So thanks to everyone.